Hello, Mr. Allman here, and this is your HL Allman cast on photosynthesis. Photosynthesis is a complex metabolic pathway, enzymatically controlled to convert the matter present in carbon dioxide and in water into carbohydrates using the energy from light. In this video, I want to give you a quick introduction into the ecological importance of photosynthesis, look at the anatomy of the plant cell in which photosynthesis occurs, and go through the two major steps of photosynthesis so that you understand a little bit about the chemistry involved. To begin, please understand that there's a reciprocal relationship between the process of photosynthesis and the process of respiration. There is a little bit of overlap between some of the steps involved in either of those processes, and most importantly, understand that the inputs of one process are the outputs of the other. Remember also the importance of plants in terms of supporting food chains. The energy from light is ultimately passed on through the carbohydrates that they make to all other organisms here on Earth. And so all the food that you eat is ultimately the product of light being converted by plants into chemical energy. You should take some time to be able to learn the chemical reactions for both photosynthesis and respiration, photosynthesis obviously being more important for this unit. But once you learn one of those reactions, the other one is just going to be the inverse of it. Six molecules of carbon dioxide, when combined with 6 molecules of water, in the presence of light energy in a chloroplast of a plant cell, is going to produce carbohydrates, which we could summarize with the glucose C6H12O6 plus oxygen gas as a byproduct. While we are focused on plants here in this unit, remember that plants are not the only photoautotrophs. There are cyanobacteria as well as algae, both of which are extremely important and provided most of the oxygen present in the atmosphere. Although land plants are the dominant photosynthesizers on Earth today, these have been around for many billions of years longer, and as such are responsible for most of that oxygen gas that's there. In plants, the primary place where photosynthesis occurs is in the leaves. And the leaf is a tissue structure made up of several different layers of cells. The cells that are specialized for the process of photosynthesis are called the mesophyll cells. Those are going to be present at the top, the uppermost layer of the leaf, and each of those cells is packed full of chloroplasts. And chloroplasts are going to be the site of photosynthesis, and we'll take a closer look at the anatomy of the chloroplast now. As you can see in this image, taken through a microscope of a cluster of plant cells in the mesophyll of a leaf, plant cells are not universally green. The greenness actually comes from the chlorophyll, which is present only in the chloroplast organelles. However, in this group of cells, there are so many chloroplasts that from a macroscopic perspective, the plant does look entirely green. The origin of these chloroplasts can be explained through the endosymbiosis theory. At some point in our distant evolutionary past, there was a eukaryotic host cell, a cell that had some but not all of these plant parts. This cell enveloped cyanobacteria, a simpler prokaryotic cell that was already performing photosynthesis and had all those enzymes necessary to carry out the process. As it engulfed it, it did not eat it, but instead kept it in a protective outer layer and utilized those cyanobacteria as an additional source of energy. Over time, these bacteria became dependent upon their host cells, and they both grew and divided together, creating the chloroplast as a permanent part of these plant cells. As we look at the structure of the chloroplast, there are a couple of parts that you absolutely need to know. First of all, understand that we have an overall rectangular shape to the chloroplast. There is a phospholipid bilayer that surrounds the entire thing. There's also a folded bilayer inside that's in a complex structure called a thylakoid. The thylakoid is going to be these stacked discs which are bound by a membrane that create these kind of tight little compartments in which materials will move in and out during the process of photosynthesis. Outside of those thylakoid, you have the stroma. So the fluid that surrounds the thylakoid is the stroma. So now we have two areas that are going to divide up our process of photosynthesis. Part of it happening in the stroma, which is in the chloroplast but outside the thylakoid, and then the part of photosynthesis that is happening inside that inner membrane in the lumen of that thylakoid itself. As we apply that now to our two main parts for photosynthesis, we start with the light-dependent reactions. The light-dependent reactions occur inside the thylakoid, so they are inside that inner membrane represented by those flattened green discs. So this is going to be where chlorophyll is located. The light-independent reactions are occurring in the stroma, the area in the chloroplast but outside of those thylakoids. These two statements can give you a good summary of what it is that I'm going to cover in a little more detail right now. You can see on the bottom of the Prezi here, thylakoid represents the location where this is occurring. So we're looking now at the light-dependent reactions. Even though I'm talking about this part first, in most plants and in most cells, these processes are happening simultaneously. The title here gives you a clue for what one of the inputs are. And obviously light is going to be one of the inputs, the energy input for this process. When light hits chlorophyll, that photosynthetic pigment inside the thylakoid, it's going to excite electrons. So as the photon of light hits that chlorophyll molecule, some of those electrons are going to gain some of that energy from the light. When they're in that excited state, they are going to move, and they can actually freely move away from the chlorophyll molecule to which they were originally bound, and are free to be passed on to other molecules in this set of reactions. This is where we go into the photosystems. There are two important photosystems to learn about, and we actually begin with photosystem 2. The photosystems are going to be a complex of molecules, including chlorophyll and other pigment molecules, embedded within the membrane of the thylakoid. So obviously we have membrane-bound proteins that are going to hold onto those pigment molecules. As the photons come in, they're going to excite electrons, and the black line shows how those electrons get passed on from one molecule to the next within these photosystems, again represented here by a protein complex which is holding chlorophyll and other parts. Those electrons are getting passed on to a final electron acceptor through a process called an electron transport chain. So the electron transport chain is going to be proteins that accept that electron but then pass it on to a neighbor, much like a bucket brigade. So on the bottom here you can see the inside of the thylakoid. On the top here is the stroma, and so this is happening on that membrane that separates those two areas of the chloroplast. Photosynthesis really begins with photosystem 2. And here the central chlorophyll works best at light of wavelength 680 nanometers. And this is going to be roughly the start of the electron transport chain. You can see this image represents the same as what you saw above, except showing you in a little more detail what it is that photosystem looks like. And you can see how it's a complex of alpha helices and beta sheets between the different proteins involved in producing these photosystem complexes. Those electrons are passed on. We have two different systems. Each are utilizing a different wavelength of light to be able to excite electrons and pass those along. Now, because electrons are being removed from chlorophyll, in order for this process to continue, those electrons must be replaced. Now, the electrons don't come back after they're passed on from chlorophyll when they're excited. So, in order to be able to replace them and allow photosynthesis to continue, photolysis is going to be utilized. Photo meaning light, and lysis meaning to split. So here, water is being split by light. And as water is broken down, we are going to produce hydrogen ions. So we have protons that have lost their electron. So those are now H+ ions. We have electrons, which will be given back to chlorophyll. And then finally, we have oxygen gas. So from H2O, we have proton, electron, plus oxygen is what is produced as water is broken down. that oxygen is going to be the byproduct of photosynthesis. It has no further use, and so it exits at the end of this chemical process. The protons and the electrons are both important. Electrons going back to chlorophyll, and the protons will have an important role next. The reason why the protons from water are necessary is to drive a process called chemiosmosis. Chemiosmosis is going to be the movement of protons across the membrane to be able to generate ATP. We start with those hydrogen ions. Again, those protons, those charged ions are going to be pumped into the thylakoid space. These small disks get filled with a high concentration of protons. That creates a concentration gradient. And based on the principles of diffusion, those protons, if given a space or a channel to be able to do so, will move by the process of diffusion from a higher to lower concentration, namely into the stroma. Now, the channel that they have to be able to do so is an enzyme called ATP synthase. Synthase being the ACE telling you it's an enzyme, the ATP synth is telling you what it's doing. It is synthesizing ATP. So ATP synthase utilizes the movement of those protons through the center of that enzyme to be able to drive together the photophosphorylation of ADP into ATP. Photophosphorylation meaning the addition of a phosphate onto adenosine diphosphate using the energy from light to be able to make adenosine triphosphate. So ADP and an additional P is combined together to make ATP. As long as there are protons, then ATP is going to be produced, and this is going to be a kind of energy currency that the cell will be able to utilize for all different types of processes to do further work. This analogy hopefully will help you a little bit in terms of what's happening in these photosystems. In photosystem 2, we have photons of light. Again, that 680 nanometer light in particular is exciting electrons from chlorophyll. Those excited electrons are being utilized to make ATP. And ATP, again, is a source of energy that will be beneficial later. Those electrons can get excited again by 700 nanometer, particularly by 700 nanometer wavelengths of light, to be able to excite those electrons again and to produce NADPH. NADPH is an electron acceptor. It's going to hold, represented here by this bucket, hold those electrons for use by a sister process in photosynthesis. So just to summarize, the inputs here, we have light as a source of energy, we have adenosine diphosphate and a phosphate which will be combined together, we have NAD+, which is going to be reduced into NADPH, and we have water, which is a source of protons for NADPH. We have the oxygen from water will form O2 as a byproduct, and we have the electrons from water which will go back and replace the electrons that were lost from chlorophyll. Our outputs are ATP, NADPH, and oxygen gas. That brings us then to a second major process in the process of photosynthesis, and that is the light-independent reactions. First thing to note is that the light-independent reactions, as it suggests, don't rely upon the energy from light, but they do rely upon the energy from the molecules produced from the light-dependent reactions. So they are actually happening simultaneous with and do rely upon the energy that is converted from the light-dependent reactions. A synonym for the light-independent reactions is the Calvin cycle, and we'll learn a little bit in a minute about why it is that it's called the Calvin cycle. It has three main phases, starting with carbon fixation. So carbon fixation is the process by which carbon is fixed to larger carbohydrate molecules. The source of carbon is carbon dioxide. So you can see CO2 is represented by the molecule here at the top. The blue line shows that it's being combined together. We have RUBP, ribulose bisphosphate, the structure of which you don't need to know, but the name of which is important. The abbreviation would be fine for you to learn. Now there is an enzyme called ribulose bisphosphate carboxylase, which you can call Rubisco for short. Rubisco is going to be the enzyme that mediates the combination of carbon dioxide with RUBP. RUBP is a 5-carbon molecule that when carbon dioxide is added, now it will become a 6-carbon molecule, which is immediately split into 2 molecules of glyceraldehyde 3-phosphate, which is G3P for short. So Rubisco combines together a 5-carbon and a 1-carbon molecule to form 2 3-carbon molecules of G3P. One of those G3Ps can be utilized by other metabolic pathways, for example, those that will make carbohydrates like sugar and cellulose. The other 3 G3P will proceed through the rest of the cycle to be recycled back so that the Calvin cycle can continue. The reduction phase, that process by which the splitting happens, requires both ATP and NADPH. So that's an energy-consuming process. ATP and NADPH both came from the light-dependent reactions. And so here those energy sources are immediately being utilized to be able to make these G3P molecules. Now, ATP is then going to be used again to be able to reconstitute RUBP from G3P. So we have many more steps represented by these black lines, the specifics of which you don't need to know, but you should know that G3P is going to be converted back into RUBP using the energy from those 2 types of molecules. Another note on terminology, reduction. Reduction is a chemical process. It is a process by which molecules are going to gain electrons. So remember the acronym oil rig. Oxidation is loss of electrons. Reduction is gain. Oil rig. Reduction is gain in electrons. Now, sugar is not a product here. So even though at the beginning of this, I told you that that C6H12O6 glucose is a product or an output of photosynthesis. And even though that is commonly utilized in the equation for photosynthesis, it's technically not a product of the Calvin cycle. G3P is the final product. G3P is a kind of simple carbohydrate containing carbons, hydrogens, and oxygens, and a phosphate group. And it can be combined together to form sugar molecules, and as we combine them together and form polymers, they can form polysaccharides. So G3P is the actual technical product, but that can be used in separate processes to make sugar. So the reason we call this the Calvin cycle is because of Melvin Calvin, and this is the scientist who designed an experiment that actually determined the molecules that are in the steps of the Calvin cycle. The way he did this is he used radioactively labeled carbon atoms, because we know that carbon dioxide is utilized during the process of photosynthesis, and we can chemically separate the different types of carbon-containing molecules that are found within chloroplast at any given time. But what he wanted to know is what is the sequence, what was the order in which these molecules were produced. And so in this is called the lollipop experiment. The picture here of the actual equipment is shown above. He had this specially designed piece of glassware into which he would put radioactively labeled carbon dioxide molecules. So the carbon in that carbon dioxide, he could trace through and follow it as it was used in the Calvin cycle. So he would inject that carbon dioxide into a flask containing photosynthetic algae. Then he would shine light on it to allow the process of photosynthesis to occur. And after a specific amount of time, he would drop the cells in which photosynthesis was occurring into boiling alcohol, which would immediately stop the reactions and kill the cells. Then he would use paper chromatography to be able to show where it is, what molecules those radioactively labeled carbon atoms were currently found in. And so what he found was that G3P, shown in this diagram as 3PGA, that's an alternate name here, was that G3P was the first molecule that was produced, because he would shine the light, and then for a given period of time, like 1 second, 3 seconds, 7 seconds, 15 seconds, he would progressively allow more and more time for photosynthesis to occur, allowing additional steps of the Calvin cycle to occur, and then do the look at the radioactively labeled CO2 and see where that carbon atom was currently located. As he repeated this process over and over for different time periods, he actually traced through and found what the final resting point for that radioactively labeled carbon actually was. To summarize here, the inputs for the light independent reaction per G3P, we have 3 CO2 molecules, 9 ATP, 6 NADPH, with an output of 1 G3P, which will go back into the, which can be, utilized to be able to make carbon-containing molecules like glucose, and then some spent ADP and some spent NAD+. Those molecules can then be recycled back in the light-dependent reaction, and they can be phosphorylated and reduced again so that this process can continue. This is a common theme that we see through these complex metabolic pathways, is that we have large complex molecules that will be reused as they get cycled back and forth between 2 different stages. This is necessary to be able to save a lot of time, effort, and energy of the cell, so it doesn't have to resynthesize some of these complex molecules and waste that energy doing so. So again, we have the light-dependent reaction, where light is utilized to make ATP and NADPH, and then the light-independent reactions happening outside of the thylakoid in the stroma, where carbon-containing molecules, which will become carbohydrates, are actually synthesized in the process of carbon fixation. With that, our summary of photosynthesis is complete. Please let me know if you have any questions. Thank you for listening.